Hello, everyone, and welcome to today's live broadcast, Transient Expression High Cell Density Culture for Increased Protein Production. I'm Julianne Chiat of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar sponsored by Gibco, part of Thermo Fisher Scientific. For more information, please visit thermofisher.com/proteinexpression. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the window. If you have any technical problems viewing or hearing the presentation, please click on the support button at the top right of the presentation window or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speaker, Dr. Puneet Khandelwal. Dr. Khandelwal leads the critical regeneration group at the Teva Pharmaceuticals. His group has focused on cloning, expression, purification, and characterization of recombinant proteins, and evaluating and establishing several platform mammalian expression systems. Dr. Khandelwal has vast experience in molecular and cell biology and is the recipient of several fellowships and grants. He's received his doctorate from Jawaharlal Nehru University, India, and completed postdoctoral fellowships at the University of Pittsburgh School of Medicine. I would now like to turn it over to Dr. Khandelwal for his presentation.
Good morning, everyone. Before I start, I thank both Thermo Fisher and LabRoots for organizing this event. I think it's a very nice platform where vast variety of people can interact and learn. So the title of my presentation, is about transient expression, how the high cell density culture can increase the protein production. And before I go into my presentation, I'd just like to tell you that unlike some other presentation, this is not going to be like a single story. So what I try to do is to capture some of the cases, some of the challenges that we faced while implementing, some of the high cell density culture. So you will see a lot of snapshots of some of those cases, and I'm happy to answer your question if you have anything specific for any of those examples. So before we start, just to give you a brief, historical background, obviously, recombinant proteins are currently produced in biopharmaceutical industry for various uses, which includes, obviously the biggest one is therapeutics, vaccines, as well as reagents, which could be part of a kit or could be to support bioanalytical assays. In last, I would say, a decade or decade and a half, a mammalian-based expression system supports more than 80% of the therapeutic protein. And one of the key reasons is obviously its close resemblance to native protein, both structurally as well as through biochemical properties. And you can understand this is a prerequisite for a safe and successful therapeutic candidate. Further, the production of reagent for bioanalytical assays, where it could be, a large classes of protein, requires a fast turnaround period for supporting both preclinical and clinical studies, including post-marketing commitments. And this is where we believe that a transient gene expression system has a superior function compared to the stable cells. So, when it comes to mammalian-based expression system, you can broadly separate them into two categories. Stable cell lines, which is, I guess, is the go-to for all manufacturer level production. But then you also have the transient gene expression systems. One of the key advantages of these transient gene expression systems, they provide a rapid method for production, thus fueling the need for reagent generation, as I mentioned, for various assays, as well as supporting robust discovery and early stage development. And if you look at the literature in last, about a decade or 15 years, there is a significant improvement in transient gene expression, resulting in gram, or sometimes, there are reports to more than a gram level of production. This could be due to either using improved expression vectors, media, certain selections, as well as cell lines. And when it comes to cell lines, obviously two cell lines are becoming a go-to cell line for pretty much everyone in the industry, which could be either a HEC-based system or a CHO-based system. And there are several different high cell density system that came up in last I would say few years, and that aided the generation of these recombinant proteins at a much higher level than we had previously seen. And one of the such system is XP system from Life Technologies, whether it is XP293 based on HEC293 cells or XpiCHO based on CHO cells, that actually showed a significant increase in a consistent protein production as well as the quality of the produced material. So before I'll show some of our results on the system, if you step back and think, what do you expect from a high cell density culture? Obviously, it should have higher number of cells per ml of culture. The other key thing is it should be easy to scale up, from small plates, flasks to large volume in liters. Ideally, the cells should be non-clumpy, a single cell phenotype, and I think they should have a short doubling time, so that allow us to produce these recombinant protein at a brisk rate. When you have a higher cell number, you want a higher transfection efficiency. So obviously your transfection reagent, all other tools should also be fine-tuned, and you should be getting a consistent product quality and performance. And this should be similar to some of the earlier conventional suspension transgenic systems, which could be freestyles or EDNA-based systems. And one of the other things that we are interested is that we don't want a high cell density culture system which is very stringent. So it should have possibility to improve expression condition for recombinant proteins, particularly difficult to express protein, and which could be as simple as manipulating the temperature, adding some feed or media, improving cell viability. Can we do something in the transfection reagent and all those things? So that's another key factor for a high cell density culture. And in all, when you have all these wish lists, it should still maintain a robust protein generation to meet tight timelines. And again, this is where I strongly believe transient gene expression system has a superior role compared to the stable cells. So the XP system actually, answers or provide most of this wish list. And we implemented, a few years back, XP293 and last year XpiCHO very successfully. And this is just a snapshot of almost more than 30 proteins of ranging from several classes that resulted in at least tenfold or higher increase in expression. And this increase in expression is actually the final purified numbers. These classes of protein can vary from FC receptors to viral proteins to antibodies to half-life extension project, where we are trying to extend the half-life of protein either by FC tag or serum albumin tags. And in all the cases, we see a significant increase in the production using these XP system. At the lower half of the slide, you can see certain examples of increase in expression, and that has been tested using different analytical tools. And one of the things that I do like to clarify here is that when we did all these comparative studies between XP systems and other transient system, it's not that we use the condition of XP system and then try to implement those on the other system. We improved the expression condition to the best of our abilities in the conventional regular cell density cultures, whether it is freestyle or EBNA, and then compare that with the optimized condition of XP293. So basically, this data represent the optimized condition expression for both the system. And the take-home message is the XP systems provides a large significantly higher amount of protein compared to other systems. But this implementation was not straightforward. Like any other system, we had to face several challenges, which could be something as simple as scaling up from small flasks, 10, 20, 30 ml to larger volume, liter or above. How to manage the cell passing, because both XP's systems, they have a very fast doubling time, particularly CHO cells, where it's about 18 hours. We tweaked a little bit with the transfection protocols. In a lot of cases, we see a significant increase in the production, but that gets complicated because a lot of that material went into the aggregates or higher order oligomers and clipping. There were always some concern about cell viabilities when you're growing such a large cell mass. And in certain cases, and I'm going to discuss it later in my presentation, particularly with the CHO system, we saw a pattern of shifting molecular weight and how the band actually looks like on a routine STS page. So we face all these challenges. We try to answer some of these things, and like I mentioned, I think going forward from here, a lot of my slides will be the snapshots of some of the improvements that we were able to achieve. So the very first thing that we faced is with, and I think that this is the XP293 studies, is the scale up. So we were very successful implementing this system at lower volume and all the way up to hundreds of mLs of culture. But when we try to increase the production level to liters, we saw a significant dip in the production. And one of the way we answered is actually by manipulating the rotating speed or shaking speed of the culture, and that significantly improved the expression at a larger volume, and it's as comparable as when you're getting the small flask studies. So the idea is that it should be kind of replicated of what we are seeing in small flasks, and we want to go to all the way to larger volume. And we are now able to do a fairly similar level of expression starting from 1 ml culture to up to 3 liters. The other problem that we had is and this is more with 293 again, is the transfection protocol. So the original method that comes from Life Technologies suggested a 3-day transfection, where you are basically seeding the cells on day 1, you're performing the transfection on day 2, and then you're adding enhancers, which one of the key elements for the increase in production. But the trouble with us is that limits the number of days we can actually do the transfection. So essentially, you have to start by Wednesday, if you think about a 5-day week, to finish the transfection. And that sometimes is not ideal the way we wanted to do these studies. And the other problem is it also kind of getting a little tricky how we want to divide our cells, because either we pass the cells on Friday on a low cell density number to stretch them to up to day 5 on Wednesday, and then on Tuesday, and then prepare them for transfection, or alternatively we do a second passing on Monday. So it's just an extra steps for us. So what we did, we actually use in-house lipid-based transfection method, and add some additives, and by making these changes, we were able to get similar level of expression that we were getting from using XP-fectamine, but reducing the transfection protocol to now 2 days, and that allows us to do a lot more things and flexibility compared to a 3-day protocol. So the other thing is, like I mentioned, in a lot of cases we start seeing a lot more protein, but a lot of these protein is getting aggregated, and then it gets very hard for the downstream people to do the purification, because like in this case, this is a case of an FC receptor. We did not see much of the expression in any of the other transient system, and frankly, the XP system showed the expression for the first time. But like you see in the in the western blot towards your left, a lot of that protein is actually aggregated. So that's not helpful. So what we did in these, in this case, we optimize the expression condition, and that essentially, playing with the temperature. So how, when you do the transfection, you move the culture after a certain period of time at a lower temperature, and that significantly, improved the quality of the protein. As you can see that almost all the protein is now, in the desired monomeric forms, and you're not seeing any of these aggregates. So again, this is very useful with this system, that it allows us this kind of flexibility to move around when it move around with the expression condition to get the best to get the right protein isoform. The other problem we faced in several cases is the clipping. Again, you see a lot of protein, but some of it is forming aggregates and some of it is getting clipped. So again, by shifting the expression to a lower temperature and adding some protease inhibitor, significantly improve the quality of the protein. And you can argue by comparing lane 1 and lane 2 that the total production in lane 2 is less than the lane 1, and we accept that. But this is basically a Western blot of a day 2 culture harvest, and by manipulating the temperature and adding the protease inhibitor, we increase the quality of the protein, and that allows us to push the production to all the way up to day 4. So at the end of the day, when it comes to the total purified protein of the desired form, the levels were fairly comparable. So that's the take-home message. This is a very interesting case. So this is a protein which is in Halflife, which is part of our Halflife extension project, where this protein was tagged with a serum albumin. And when we initially tried to check the expression, and this is basically size exclusion HPLC for the culture harvest, a significant portion, I would say almost 60–70% of this protein is going into an aggregate forms, which you can also see in the very top of this STS page, lane 1 on the right hand side. And this was a challenging, because we really wanted this project to move forward. So obviously the very first thing we did is optimize the expression condition, again essentially playing with the temperature, and then see if that improves the quality of the protein and reduces the aggregates. It impacted a little bit. You see now the aggregates, I would say maybe like 50–50 or something, which is also very clear in the STS page on the right hand side. But it's still not like it's still not reached to the ideal stage where the downstream feel comfortable to purify this protein. You still have a significantly large amount of aggregates, which, it's harder to purify. So what we did in this case, we not only play around with the expression condition post transfection, we also manipulated also manipulated how we are performing the transfection. We changed our vector to a slightly weaker promoter. We manipulated the DNA concentration and how we are mixing the DNA and the lipid. And in all, there was several optimization. And once we implement all those things, we reduces the aggregates to almost 20% or less. And you can see that the monomer expression also increased considerably. So again, this comes back to the flexibility of this system, that it is not very rigid. So it allows you to do several improvements to express some of these difficult to express protein. And I think for this protein, the final purified product that we got from a transient system is about 150 mg per liter. We needed about a gram total protein to carry some of our studies, so we were able to achieve that level. And like I mentioned, we reduces the aggregation by almost 80% or above. So it was a very exciting result. And one of the other key factor, when we were making all these changes, by the transfection conditions or by manipulating how we are doing the transfection, we saw, that allows us to. What we observed is that there is an increase in the cell viability. In the picture here on the left-hand side is an experiment performed, using the manufacturer's recommended conditions, and on the right-hand side, it's our optimized condition, and you can see that there is no other way to show you the cell viabilities, except showing pictorially the cell debris, and we were able to increase the cell viabilities to day 7 or day 8 in XP293 system. And in terms of production, that helped us to increase the production somewhere between 10 to 20%. Obviously, that depends on case-to-case basis or protein to protein. But again, this is one of the challenge we had, and we improved the conditions. So the next few slides, I'm going to just compare some of the different classes of protein which we picked and compared for the expression of XP293 and ExpiCHO system. Obviously, like I mentioned, XP293 has been here for quite a number of years, and ExpiCHO has been launched only last year, so I think it's still evolving. But we want to see how it performs for some of our proteins. So this is an example of an IgG expression. And before I go into the results, I must say that the ExpiCHO, the way it come out, it actually allows you to play a little bit more in terms of the expression conditions. So the LifeTech recommended doing expression at 2 different temperatures, and also there is an option to add feeds to increase the productivity. So the way we perform these studies is we did the transfection as per the recommended protocols by LifeTech. We did not do much changes when it comes to ExpiCHO, and then we followed the expression at 2 different temperatures, and we included the feed steps as recommended by the LifeTech. And so the 2 temperatures here are 32 degrees Celsius and 37 degrees Celsius. And essentially, lane 1 to 6 are the samples collected for ExpiCHO at 2 different temperatures, 1 to 3 at 32 at days 6, 10, and 14, and 4 to 6 are temperature 37 at days 6, 10, and 14. And lane 7 is our ExpiCHO. Again, for a lot of these proteins, we already made some improvements or whatever best we could do. So we just picked that condition. We did not want to do the whole time course. And as you can see that at both the temperature, whether it is 37 and 32, ExpiCHO showed a significant improvement for IgG production. And in general, we tried several IgGs, and ExpiCHO showed improved expression. The expression improvement could be as low as 2-fold to as high as 15 to 20-fold. But I think except one case where it is not much improvement, on all the tested antibodies showed an improved expression using the CHO system. Then we also tried using some small molecular weight protein. So this is a cytokine which is on the lower end of the molecular weight of 20 kilodalton or something. And if you look at the box, you can see that again, if you compare the expression, ExpiCHO seems to be doing better. But unlike IgGs, where if I go back to my previous slides, where if you look 1, 2, 3, you continuously see an improved expression. Same at 4 to 6. For this protein, it's a different pattern. Where at lower temperature, if you just have your attention on lane 1 and 2 and 3, you can see a continuous increase in expression as we are going further along in the incubation time. However, on the other side, at 37 degrees Celsius, you start seeing that it peaked around day 10 or day 10, but then it kind of goes down. So that's kind of interesting for this protein. The other thing that we observed in this case is the shift in the molecular weight, how these protein runs in two different systems. So like in ExpiCHO, they're running at a slightly lower molecular weight compared to XP293. And then I'm just trying to remove these red lines, but anyway. Coming back to the point that ExpiCHO produced material is running at a slightly low molecular weight compared to XP293 produced material. The other thing that we observed is the compactness of the protein, where the ExpiCHO produced material ran a slightly more compact band compared to XP293 produced material, which is more diffused. We don't have any glycosylation data for these proteins to realize whether it is due to difference in the glycosylation or it is something else going on here. We do know that the XP293 produced material works very fine in our assays. We haven't tested the ExpiCHO material, so that's something we need to check. And this shift in the molecular weight or this shift in the band pattern we observe for some other classes of protein. And this is again another example of an Fc receptor. Again, if you see, if you compare lane 1, 2, 3, you start seeing a gradual increase in the expression at 32 degrees Celsius. When you compare it with 4, 5, 6, in lane 5, you see the highest level of expression around a week and a half, and then it kind of top off. The other difference is again, how these proteins are running on a gel from XP293 versus a CHO system. But one other interesting thing about this protein that we found out is that there is, if you just look within one temperature range, there is a difference how the protein behaves. So, at early stages, it ran at a slightly higher molecular weight, and then it kind of settled down at a lower molecular weight. And we saw the same pattern at the 37 degrees Celsius also. So again, we don't know why this is happening, but we observed this kind of phenomena for some of the proteins. And right now we are trying to do some functional studies or doing some affinity measurements to understand this difference in the running as well as band pattern. Excuse me. This is a very interesting case for some other Fc receptor. So what's interesting here is that the expression level of this protein in both 293 or CHO were fairly consistent and very similar. But when we did the purification, involving two steps, first the affinity column followed by gel filtration column, what's exciting or what's very intriguing about this protein is it has a very different dynamics when it comes from CHO versus 293, where 293 produced material on your left hand side showed a significantly large amount of aggregate population compared to the CHO or ExpiCHO system, where you see a large monomer population compared to the aggregate. So we had seen this case only for one of our protein, but this is again very intriguing. And what we like about this system is that it gives us a flexibility from case by case. There may be cases where the protein is good in one system in expression wise and may not be good in the other one. So it provides us all these different flexibilities and different options to try and get the material of interest. I think with this, I will go briefly into my conclusions. We believe that both ExpiCHOs and 293 high cell density cultures are superior and produces 10 or higher fold of protein than the conventional suspension culture. Like I mentioned, the expression behavior is empirical to each protein when you compare T93 versus CHO cells. Again, both the system allow us to optimize the expression level and to improve the quality of difficult to express proteins. Like I mentioned, that the shift in the molecular weight needs to be evaluated, whether it is due to some post-translation modification or there is actual some variability in function. Those are the studies that are ongoing. But in all, both these systems produce consistent product quality and behavior and maintain a robust timeline. And particularly, this is a very important aspect for our group, because we support, a lot of reagents which are critical for bioanalytical assay development and analysis that supports, the preclinical and the clinical studies. With this, I like to conclude my talk, and I do like to acknowledge, my manager Rita, and my second level manager Dr. Lou, and everybody in the team, for being very supportive, doing all these analysis and doing day-to-day activities. With this, I think we can take questions.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You could submit questions by typing them into the Q&A box, which can be found by clicking the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, does this give us PACE credits? Oh, my apologies. We're doing another question. What is the temperature adjustment that you use to improve the protein quality to prevent protein aggregation?
Can you hear me? Okay, so most of the temperatures were done are, the temperature has been moved towards the lower side of 37 degree. The key point is at what time point you want to move the culture, once you do the transfection, and that is a little bit protein to protein specific. I don't think we have a generalized number. We do have some general condition that we try first, just to see if it is improving the expression or not. But for optimization, you need to play around a little bit, whether you want to shift the flask within few hours of transfection, or you want to wait for a day and then move towards the lower side of the temperatures.
The next question, to increase protein production in high cell density cultures, have you used hollow fiber bioreactor?
Not yet. We haven't done that, so we don't know how these high cell density culture will behave there. We are still using conventional flasks. Now you can get up to 5 liter flask supporting 3 liter volume culture, so that's what we predominantly use.
What is the shaking RPM from small scale to liter scale?
Okay, so the small scales are around 125 plus minus 5, and when you are going to liter scale, you have to bring it down to somewhere between 90 to 100. And I think, if I remember correctly, the ExpiCHO manual actually gives you a formula also to calculate the RPM. And obviously this also depends what kind of throw speed or orbit diameter your shaker has. So, my orbit diameter might be completely different from your shaker orbit diameter. So those are some of the things that you need to optimize.
What is your typical go-to expression volume, and how is it determined? Are you working towards a higher throughput format, negative 2 milliliter over 50 milliliter?
So a typical go-to expression volume, I don't know if you're in what context you are asking, but if you are doing the initial evaluation, it is in the range of tens of ml, and we do wanna go towards a higher throughput format, and that's what we are doing, not over here, but in our another department for the antibody discovery group, and we implemented this in a high throughput format and tens of ml of culture.
If there are no more questions, I would like to once again thank Dr. Khandelwal for his presentation. Do you have any final comments? One more question just came in. What's the highest transfection cell density you have tried?
So I'm not sure you're talking about which system, 293 or CHO. With the 293, and CHO, I think in both the cases we haven't manipulated the cell density. We just followed the recommended cell density by the LifeTech, and obviously for 293 is 2 to 2.5 E6, and for CHO is about 5 E6. So we haven't played with those numbers. That's something we can try, but we haven't done those studies.
Okay, so now there are no more questions, and I would like to once again thank Dr. Khandelwal for his presentation. Do you have any final comments?
No, I think it's. I'm hearing some of the other talks. It's like I said, it's a very exciting platform, and it's. And I thank both, Thermo Fisher and Lab Roots for bringing me in and introducing me with all of you. And hopefully we will have something similar in near future to share more. Thank you.
I would also like to thank Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. For more information, please visit thermofisher.com/protein expression. Today's webcast will be available for on-demand viewing through May 4th, 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.